hi everyone. I'm happy to welcome you to our monthly series of webinars with the Stilla Technologies. My name is Alexandra Aboud. I'm Senior Marketing Manager here at Stilla. Today I have the pleasure of introducing our speaker, Dr. Jörg Tost. So Dr. Jörg Tost is the Director of Laboratory of Epigenetics and Environment at CEA-CNR GH Évry, France. His laboratory analyzes epigenetic modifications using state-of-the-art technologies and develops bioinformatics tools to process such data, aiming to implement epigenetic modifications for personalized medicine. The research focuses on epigenetic changes in cancer and inflammatory diseases, the relationship with environmental exposure, and the development of novel technologies for liquid biopsy analysis to improve treatment management. Dr. Tost is the author of more than 246 publications, a senior editor of the Journal of Epigenomics, and is involved in several international consortia, including ICGC and Rescuer. Today, he will talk to us about microRNA signatures in clinical practice, the development of multiplexed microRNA analysis with digital PCR. We will open the floor for questions at the end of the webinar. You can submit your questions under the questions tab on the top right of your screen. With that, please join me in welcoming Dr. Jörg Tost.
Thank you very much for the kind introduction. So for us, really, the development of this multiplex microRNA analysis by digital PCR is a major step forward. And I let you enjoy my presentation, which I did at the Festival of Genomics a few weeks ago, where I describe in detail this development in collaboration with Stilla. And then if you have any questions, please reach out and I will hopefully be able to answer any questions you might have. Enjoy. Okay, thank you very much. So I will shift gear a little bit and I will talk about our work on microRNAs, which are, if you want, some other epigenetic modification, something which is very important as a biomarker, but we are not yet there in the clinics. So microRNAs are, of course, very important. You might have seen that the Nobel Prize in Medicine went to microRNA discovery this year. And microRNAs are interacting with translation by blocking the translation or by degrading the RNAs. So what's really important is, like any other epigenetic modification, microRNAs are cell type specific. So each cell type has its own microRNA profile. If you profile microRNAs and PBMCs, this will mainly depend on what cells you have in there, what you will see. So what we do is, in most of our studies, we will only look at sorted cells by FACS or by MAX to have a pure epigenetic or microRNA profile of a given cell population. So you can do this, of course, in tissues, but most people are nowadays interested also in liquid biopsy, because microRNAs are something which is physically very stable. You can actually analyze microRNAs in samples which have been stored for decades. You can analyze them in plasma, in serum. The profile is not the same, depends a little bit on what's in there, but also in extracellular vesicles or exosomes, which are very different from what is circulating. Circulating is mainly determined by cell death, while exosomes is an active process of signaling. If you look at the literature in microRNA, there are each year thousands and thousands of papers on microRNAs. And if you look at those which actually use microRNAs as a biomarker, you will see that approximately 1 out of 4 papers is using microRNA as a biomarker. So you have 5,000 papers on biomarkers each year. And, well, in the clinics, there is hardly anyone. Why is that? So one of the best studied microRNAs is miR-21. You might have come across that. And it has been proposed as a very good biomarker for colorectal cancer, and then for breast cancer, for liver cancer, for renal cancer, for any cancer you want. And it has been a biomarker for allergic diseases, a biomarker for multiple sclerosis, and a biomarker in nearly any neurodegenerative diseases. So if you do the list, I mean, I didn't do it, other people did it. It's actually a biomarker for all diseases which are known, more or less, which means it's not a biomarker at all. It lacks a complete specificity. So it detects something, but you don't know what. And it's also, of course, associated with a number of quantitative traits which have nothing to do with disease. So of course, people were realizing this and that this is not bringing us forward. So people started to work on signatures, combining a number of microRNAs. So I only will show a few examples which are more or less randomly picked based on the criteria that they have been replicated in several clinical trials already. So, because if you take only signature, again, you will be in the thousands of papers. So for example, the MicroRNA signature will predict early relapse in epithelial ovarian cancer, and this has been shown in several papers. And another application is using a microRNA signature as a negative predictor in ambiguous lung cancer or computed tomography for lung cancer detection. So actually what you do is, if your microRNA signature is negative, you're quite sure that there is probably no lung cancer in those. If of course, CT is positive, there is no reason to think there is no lung cancer. So it's only the negative predictive value which is of importance for this. How do we analyze microRNAs? Well, two main techniques. Either we sequence, easy, a lot of data, very fast, but there are some problems. Because sequencing is based on ligation protocols, it's not an absolute truth representation of the distribution of microRNAs. What you can do also is, well, qPCR, that works tremendously good. You can use, depending on the supplier of your preference, either CyberGreen, LNA enhanced, not LNA enhanced, TaqMan advanced, whatever you want. They work relatively good with some differences, but these are normally to be done one by one. For signatures, especially in liquid biopsy, this can be problematic. So a relatively, well, a younger technology, I shouldn't say new technology anymore, is digital PCR, which is very simplified. You create partitions based on physical separation, oil or plastic, and you will have one microRNA, one target molecule, one mutation in one of these wells or droplets. So you then do as sequencing, you just count your positive droplets for your target, in our case, microRNAs. So what really has been a quantum leap forward for this is that several suppliers have provided multi-channel digital PCR experiments. We have decided to go with the technology from Stilla, which has in this version 6 channels. They now have a 7-channel instrument. So you use 6 fluorescent channels, and you can thus target 6 different microRNAs in one readout. You will scan each color separately and then get these nice droplet plots on the right. So the workflow is really simple. You just pipette in your reaction, in our case, an RT reaction because it's RNA. You pipette this in. So Stilla works a little bit different than some other droplet providers because actually the droplets are formed in their what they call sapphire chips. So actually you don't have to handle the oil. This creates the droplets just by the size of the channel. You cycle until you're done and you put it on the scanner. This is an image of the chip with the holes are just the separation between the tops, and you go for your 6 fluorophores. So one of the main points we had to decide on how to do this, there are 3 main principles how to do the RT reaction. Stem loop primers, which are also used by a commercial supplier for their microRNA assays. So which actually uses a stem loop primer, or which is then prolongated, which gives you also the space to position 2 primers and a probe on a microRNA. Because remember, microRNA is 22 to 24 nucleotide in length, and you need to put on 3 primers, each 20 primers. So that's slightly difficult. So the other idea is to do a poly A tail. Then, of course, you can use a universal primer on that. It's also used by a commercial test, or you can use just a polyadenylation protocol without the ligation step. So all 3 protocols are valid, and actually you can buy them if you want. But remember what I said in the beginning. So we were a little bit allergic to the idea of ligation because we know this induces some biases. So we excluded the second one. And the polyadenylation is okay. It works wonderful if you have a simplex assay, but we were afraid because we have to position the probe partly on the poly A, and actually we lose perhaps some specificity. And there has been discussion that these assays might not distinguish between pre-microRNAs and major microRNAs, which means we went for the first solution. So as I said, you take the stem loop primer, the 3' end overlaps with the 5 nucleotides of the microRNA, so it's each stem loop primer is microRNA specific. You do the reverse transcription, you get your cDNA, place a forward primer which has a non-annealing tail to have the space to have the right annealing temperature, the probe, and the universal primer, which is always just in the stem loop. So the idea was to take 6 microRNAs, and I have to disappoint you. These microRNAs have no biological meaning. It's just they're expressed in a THP1 cell line, which is our pet cell line in the lab. So no biology, just technical. Do the stem loop and then go. And all this project has been done by Florence Bisato, which is a research engineer in my lab. So we first looked on, do the constructs working on synthetic templates. So you generate the RT template. No, you buy it, or you take synthetic microRNAs. You do this by qPCR, and we actually could show that the amplifications are working very nicely. So we could go to digital. And also, what's really nice about the Stilla workflow is you can actually recover your droplets after you have done the entire reaction. So you can run a gel electrophoresis once your digital PCR is over. So actually you put this onto a bioanalyzer, or a LabGX in this case, to look at the size. And the 3 peaks, if you ask, these are the 2 primers and the probes, which of course, because we didn't purify, are still in there. And there might be a small peak for a stem loop primer. So we spent time optimizing conditions for each assay, primer concentrations, also because we were not really a digital PCR lab until this project. And what you can see on the left side, the amplitude of the signal is mainly determined by the quantity of the probes you use. So the more probes you put in, the higher the signal. And the separation is determined by the ratio of the different primers one to each other. So the details are all in the paper if you want the exact primer concentrations. But except for the third condition, where actually on the bar chart you see no signal, probably we put so much primer in that it's saturated. What we see is a lot of the reactions gives actually a very reproducible result, which prompted us to go for dilution series. And for those who are familiar with digital PCR, it's no a big surprise, but you see a very nice linear correlation also with only 3 data points. One should remark at this stage that actually the expected and the detected concentration are not exactly the same. They are lower than what you expect. Digital PCR gives you absolute numbers, only if you have no other reaction step in front of it. We need to do the RT reaction, and we all know that RTs are more or less stable and do induce some variability. So this worked quite nicely. So we went to a sixplex again. So each color separately, and the last bar is the 6 colors mixed. Again, we see what we wanted to see. And if we go for dilution series, you can already estimate from the droplet plots that you actually get a very nice correlation, which is the case. Very nice linear relationships. When you look a little bit more specific at this, you see that one of the microRNAs, which is microRNAs 335, is actually standing out. The signal is much stronger for this microRNA. This could be complicated in a biological setting because you might run into saturation. So what we figured out is actually by reducing the stem loop primer, so you only look at the green bar, it goes down if you lower the stem loop primer proportional to the amount you lower it. So actually, if you do put in 10 times less, you actually get 10 times less droplets. It's linear, and it brings then the range of the green bar into the one of the others, so they all have similar performance. So as I said, okay, we don't see what we are supposed to see. There's a difference between expected and detected concentration, but do we see, can we reproduce changes? What we did is we took a lot of different concentrations, always divided by factor 2, 2000, 1000, 0, 500, and measured the absolute concentration. And as you can see, you actually see exactly that. The numbers are not what you expect to have, but it's always a doubling of the positive droplets. So one of the other things we looked at is, are the things reproducible, especially because we need to do the RT reactions. So the 3 different colors are 3 independent RT reactions. And you see that you get mostly the same results. So if you look at this as a coefficient of variation, you are, for 4 out of the 6 microRNAs, you're actually below 5%. And for the other ones, which are those which are highly expressed, you are between 10 and 14%, which is very similar to what people have seen using simplex digital PCR experiments. And if you are familiar with the coefficient of variations you normally see with qPCR, they're easily 25%, and sequencing for lowly expressed ones might be even higher. So we were very happy with that and moved on to the cell line where we should detect it. And we used 2 different types of samples. We used total RNAs, but we also use what we call enriched microRNAs. So in the extraction step, we use actually a modified protocol, which will only give us the small RNAs, which is much more cost effective for sequencing. And what do you see, for example, for LED7A, which is the turquoise, the middle one, you get into full saturation, not very good. But of course, if you lower by a factor of 30, sorry, you get out of the saturation again. So again, the 6+ was working. So as I said, we don't work on blood. We sort everything. So THP1 is monocyte. So we went for human monocyte samples and looked again at the different samples. As you can see, again, on LED7A, turquoise is very highly saturated. So we had to adapt the assay. But as I told you before, like for my microRNA 140, well, you just need to adapt the stem loop primer and it goes down. And this is actually what we tried. And you bring now the LED7A, which is the lowest line. So it's a very low line, but it's so highly expressed that actually you reduce the signal down to a measurable range. All this was done on a platform called the Sapphire chips, but they are not really ideal if you want to analyze a large number of samples. So there's also a second product, which is called the Ruby chips, which actually have 16, so 4 times more than the Sapphire chips. But you can see, taking the exact same RT reaction, it just works. We didn't change anything. We just took the reaction from the Sapphire chip, or instead of putting it on the Sapphire chip, directly onto the Ruby chip. So again, dilution series, as you can imagine from the drop plots, this is linear, and indeed it is. So this is really an advantage, that you can change between the two platforms without any need, depending on how many samples you have. So this is a little bit where we are today. So we can go for these microRNAs, up to 6 microRNAs, but we don't think this is really the limit we have touched. There are also other multiplexing options which have been developed by Stilla, like color combinations, like universal reporters, which they call crystal flex probes. So there are different ways to look at this. Yeah. So I hope I've convinced you that non-coding RNAs are promising biomarkers. There is a big potential, which we don't use really today. But in most cases, you will need not 1 microRNA, probably not 3 microRNAs, but probably 10 microRNAs or even more. I think compared to qPCR, we have the advantage that it is relatively, or it's more straightforward. You don't need reference genes, which in our hand was always a big pain point, because reference microRNAs, because everything moves, are also not very stable. So each time we run into problems, finally going for a geometric mean in most studies, you can, for us, we can go down by a factor between 2 and 4 in sensitivity. So we know, based on sequencing, that we need at least 200 reads per million to be able to detect the microRNA by qPCR. It's about 50 for digital PCR. What I think is really nice, you can change the chip formats, doing high throughput if you have many samples, or low throughput if you want to develop or do things. I think still one of the major problems is not really the analytical readout technologies, but how you get your samples, what you have in your samples, how they have been treated, how the microRNAs are extracted. There are so many things which are often not sufficiently controlled, which hampers actually the clinical implementation. But I think digital PCR in general will make or facilitate, especially if one day there will be commercial kits for different signatures, to implement those in the clinical laboratory, avoiding some of the pitfalls we have seen with qPCR. And with that, I'm happy to answer questions, and I thank you for your attention.
Thank you for this talk and setting the stage on how to look for microRNA using a multiplexed approach with digital PCR. I will open the floor for some questions and go through all the questions coming through in the chat. I will do my best to go through these as much as possible, and then otherwise we'll reach back as a follow-up, or if you have any additional questions, don't hesitate to reach out at contact@stellatechnologies.com. So first question we have here is, considering the advancement in digital PCR technologies, how do you envision their role in personalized medicine evolving over the next 5 years, particularly in the context of epigenetic research?
Thank you for this question. So I think that we are seeing epigenetic modifications being more and more being used in clinical applications. I mean, the Cologuard tests are based on mainly DNA methylation, especially in their second generation. Of course, we have all been aware of the developments around GRAIL. These things are not yet ready, but clearly while the first generation of personalized medicine was mainly focused on genetic alterations, there is a lot of room for improvement. And I'm not saying that epigenetics will solve everything, but it's clearly giving an additional layer that in many cases can provide additional information and play a role for that. And I think, well, we are convinced that, for example, microRNA signatures, so will get the sufficient specificity and sensitivity to look at very specific questions, as other people have shown with the studies I presented in the introduction, such as the relapse in epithelial ovarian cancer or the assistance of ambiguous T scans for lung cancer. I think you need to have a very good question and then try to respond specifically to it and not just run 20 samples and find the biomarker of your choice, which in the end is not really validated. So we are convinced that epigenetics will play a major role in personalized medicine over the next years.
Okay, thank you. Moving on to the next question. Really interesting technique that you have optimized. I work with cancer therapy-induced cardiocicity and the development of potential miRNA signature cardiocicity biomarkers. We use clinical samples and we have these available for many different cancer studies and therapies. I would like to get in touch with you so we can assess miRNA panels. So yes, I'll pass that information through using different techniques versus traditional RT-qPCR. Other question, nice presentation. It seems that the multiplex works nicely for detection, but since accuracy is off, not for quantification.
Yes and no. So actually, it's not absolute quantification anymore. You lose one of the main advantages, of course, of digital PCR, but actually we use these synthetic microRNAs as a calibration curve. So what I didn't show perhaps in enough detail is these calibration curves are absolutely reproducible. So you don't, in contrast to qPCR, you don't have to do it each run. You have to do them once or once every 10 runs if you want to be sure that they are not moving or that the experimental protocol is not moving. And then you can get back to absolute numbers. So we don't need a black and white difference, so a microRNA is expressed or not expressed, to be able to distinguish two conditions. So let's say cancer detection or compared to non-cancer detection. But of course, we will have to test, and this is a project which is currently ongoing. We have to test how much difference we need to be sure that we can test the difference. So one of the things I've shown in this presentation is actually we can modulate the slope of the calibration curves by using different stem loop primer concentrations. So actually you can increase your differentiation in the region you are interested by modulating these parameters. So I would not say we cannot quantify, but rather you can quantify, but you are not sure this is absolutely the right copy number of microRNAs present in your solution.
Okay. Next question. In your opinion, what is the most critical step when designing multiplex dPCR assays for miRNA? Have you assessed the false positive rate?
So we run each assay, of course, against the other microRNAs and see that there is no signal. We also have microRNA mixtures, which is just a pool of all the microRNAs, and we make sure that we don't get a signal in those. So there are several points which are yet to be fully evaluated. This is the effect of small variations in the nucleotide sequence, but there have been previous papers in qPCR using the same stem loop PCR technology, RT-PCR technology, which showed that actually this technology is quite robust against small changes. So of course, the design is critical. The design is difficult because the microRNAs are so short that you don't really have much options to make it work. So at the moment, we have the ones we have tried to target been able to try to target one, except one where actually we got interactions between the stem loop primer and the microRNA. And this is something which was difficult to solve. There are solutions for this. There are different stem loop primers which have been evaluated and published in the literature, and it would, in theory, at least to be an option to go for those. But of course, there is always a possibility that there is interactions also between the microRNAs and the different primers, and this might lead to problems for the analysis of some microRNAs. But we have not really, as I said, in most cases, encountered those.
Okay, thank you. Moving on to the next question. What is the detection limit after RNA extraction from actual tissue samples?
So we haven't looked at tissue samples because we have been focusing mainly on blood cells, on liquid biopsy. So in general, the limit of blank is around 20 molecules. Of course, microliter. This is about the background we see. For some microRNAs, it goes down to 5. But you need, I would say, 10 to 20 microRNAs per microliter extracted to be able to detect them. But it will also depend on your, as I said, on your assay, because we see less of the microRNA after RT than we should see in absolute counts.
Okay, thank you. What do you think is the reason for the discrepancy between the nominal and detected copy numbers, especially when using synthetic miRNA mixes? Is the RT efficiency low and miRNA specific?
Yeah, so it's clearly that even if we use the exact same amount of microRNA, the RT efficiency is very different from microRNA to microRNA. It's highly reproducible. This was actually something we have been very afraid of because we have previously in qPCR-based studies identified RT as the major cause of variability. That's something we don't see with this system of RT, with the stem loop RT here, but they are more or less efficient, probably based on the binding of the overlap. So you normally had 5 or 6 bases in the end of the stem loop primer, which is complementary to the microRNA, which by default are the 3' end of the microRNA. So there's not much way to change this or to modulate this. And of course, you don't want to take too much of the space of the microRNA onto your RT primer. So there is clearly a difference in the performance. Ideally, it would be good if one could get around this RT step, but at the moment we have not found a way to directly analyze the RNAs in digital PCR.
Thank you. What is the best kit to isolate mRNAs? Do you have experience isolating circulating mRNAs?
Well, I don't think we have published a few years ago a benchmark on circulating DNA, identifying 2 or 3 kits which worked well. For the microRNAs, we personally use a kit from QIAGEN, so there are different kits for either exosomes, for circulating microRNAs, for tissue microRNAs, which in our hands work all very well. But we have not in-depth tested, and there are, for example, the miRNA kits from Thermo Fisher, which are also used by many people. We haven't tested them, so we are happy with the kits we have, but I clearly have not compared them to other kits, and I would suggest that there might be benchmarks in the literature which have tested this in more detail, but the QIAGEN kits work in our hand quite nicely.
Okay, thank you. Well, I think this closes off everything for today. We don't have any additional questions, and we'll follow up with the others. Thank you for sending through the emails, and we'll pass those on to Dr. Tost. So thank you.
Thank you. It was a pleasure, and I hope you have learned something. And if you're interested, feel free to reach out to me, and perhaps we can do some things together. Have a nice day.
Thank you.
Bye.